Hello everyone. Today we're going to be covering chapter number 5. This chapter actually talks about photosynthesis and cellular respiration. So basically we're going to be talking about metabolism. So this chapter is actually a very interesting chapter and covers a bunch of different things. We're going to start, guys, with metabolism. We'll talk about what is anabolic reactions, what is catabolic reactions, and then we'll talk about photosynthesis and cellular respiration. So today is an interesting topic as it talks about how our cells produce energy and how does plants actually produce energy from sunlight. Okay, so to start with, guys, what do we mean by metabolism? Metabolism actually talks about all the chemical reaction, guys, that are involved inside our cell. And they're involved in either making the energy and then storing that energy, and then how to use that energy when required. Okay, so that's what metabolism means. So it is the reactions that have to deal with the energy of our in our cells. Okay, so there are two types of reactions, guys, under metabolism. You have anabolism and catabolism. So you can have anabolic reactions or catabolic reactions. Anabolic reactions basically means that you take small things and then you combine them to make one complex big thing. Okay. For instance, photosynthesis is an anabolic reaction. Okay. So for example, guys, if you take carbon dioxide, which is CO2, and then you change it into a glucose molecule, which is a big 6 carbon molecule. You see you have changed a single carbon into a molecule which has 6 carbons. This is anabolic reaction, anabolism. Okay, then the exact opposite of that is catabolism. So catabolism is the breakdown of those big molecules that you have created and break them down into small molecules. And guys, this actually releases energy. Okay, so the example for that is cellular respiration. So that's exactly what happens in cellular respiration. You take glucose, and then glucose again is big carbon molecule. So 6 carbons come together to give you glucose. And then that glucose is broken down into carbon dioxide, which is a single carbon molecule. So you see you change your complex molecule into a tiny molecule. This process is called catabolism, and this guy actually releases energy. On the other hand, guys, anabolism, which means you're constructing things, right? So if you got to construct something, you have to spend energy. So that's why you can see that photosynthesis, guys, will use sunlight to produce complex molecules. So that's anabolism, that requires energy. On the other hand, you have catabolism, where we're going to use glucose molecule to give us energy, and we're going to break down glucose into carbon dioxide. Okay, all right. So after this, guys, let's quick. So this actually just summarizes anabolism and catabolism. So if you have a monomer and you join them to make a big polymer, that's anabolism, which takes energy, so it requires energy. On the other hand, if you take this big molecule polymer and then you break it down into small monomers, that's when you're going to release energy, and this is called catabolism. Okay, right. So again, as I said before, for under metabolic pathways, we have two things to cover. One is photosynthesis and one is cellular respiration. So let's see this on this image, guys. What's going on? This actually summarizes both photosynthesis and cellular respiration. And actually, you will, based on this figure, you should be able to realize that how much interdependent these two processes are to each other. Okay, so basically what, guys, happens in a big picture, if you want to see the big picture, what happens in photosynthesis is you're using the sunlight as a source of energy, and then you so by you I mean plant. So sunlight is being absorbed by the plant, and that plant is then converting carbon dioxide into glucose. Okay, so a complex molecule, right? And now that plant is making glucose for itself, so it can use it for cellular respiration. Now at the same time, animals come in and they also start eating that glucose from the plant and use that as their source of energy. Okay, so guys, plants are not making food for us. They're making food for themselves. We just happen to come in during the evolution and we're starting eating those plants and do cellular respiration to produce energy from that sugar or the glucose. Okay, so cellular respiration, as I said before, guys, is going to take the glucose and break it into carbon dioxide. Okay, and then you can see how they're dependent on each other. You guys see, photosynthesis is only going to work if there's carbon dioxide there. Okay, and which process produces carbon dioxide? Cellular respiration produces carbon dioxide. Okay, so on one hand we have cellular respiration, which makes CO2. That CO2 is used by photosynthesis to change that CO2 into glucose. Then that glucose is taken under cellular respiration and is again broken down into carbon dioxide. So you can realize how that cycle works. So one thing to remember, guys, in this chapter, in general, that plants, guys, do photosynthesis and do respiration. We only do respiration. We cannot do photosynthesis. But keep in mind that plants not only do photosynthesis. Is a common confusion among students who are first time learning biology. Plants do photosynthesis and cellular respiration. They're not making glucose for us, but for themselves. Okay, all right. So there's some few questions that you should be able to answer based on what you just learned. All right, now guys, let's talk about the actual energy molecule which is utilized by our body, which is always available to run the reactions. And guys, that molecule is called ATP. Okay, so adenosine triphosphate. So ATP is the cellular energy currency that we need if you want to do any activity in the cell. So for instance, if you have to go and buy grocery, you go to the grocery store and then you pick up the things that you want, and then you go to the cashier and then you give them cash, or you pay through your bank account. So you're paying them in dollars, right? So dollar is the currency that enables you to go and buy stuff from the market. Okay, can you go and take some other countries' currency? Okay, can you take Canadian dollars and use that in U.S. grocery store? No, you cannot. Okay, although the Canadian currency, or Canadian dollar, is a currency that has the capacity to buy things, but not in US. Okay, so that being said, that what does that mean? Why I'm giving that example? That inside your cell you don't have just ATP, but also another energy carrier molecules, like you can see those hairs, like NADPH, NADH, and FADH2. So guys, along with ATP, we also have NADPH, NADH, and FADH2 who are involved in carrying the energy. Okay, now, but if you want to actually do a cellular activity, then the actual currency that you can spend is ATP. Okay, so our ultimate goal is to produce ATP. Okay, now NADPH, NADH, and FADH2, consider those guys as an asset. Let's say you bought gold, so you can sell gold and get ATP and get dollars. Same way, you can spend your NADPH, NADH, and FADH2 to exchange for ATP inside the cell. So there are mechanisms in the cell which can convert NADPH, NADH, and FADH2 into your ATP, which can then be later used. Okay, and we're going to learn that today as well. Okay, so let's talk a little bit more about ATP. So guys, ATP, as the name suggests, it's adenosine. So adenosine is a nucleotide. We're going to learn more about that when we learn nucleic acids, when we learn DNA. You can see in the structure, guys, it's called triphosphate because you can see that there are 3 phosphates attached to it. Okay, so guys, imagine phosphates as very high energy molecules. Okay, why are they high energy molecules, guys? Let me give you the formula for phosphate. So phosphate is this PO4 with a -3 charge. You see, phosphate has a negative charge. So when phosphates are attached to each other, each one is carrying a negative charge, and that's too much negative charge right next to each other. And ATP, that's why, is considered a high energy molecule. Guys, if you have too much of charge next to each other, that molecule becomes high energy, and that's why ATP is a high energy molecule. That's why it's a energy currency used in our body. Okay, so ATP can be broken down into ADP. So here you had ATP, you had 3 phosphates. In adenosine diphosphate, you have 2 phosphates. So 1 phosphate has been taken off, and this phosphate is now by itself. So this is considered the inorganic phosphate. Okay, so phosphates can be added to ATP, and you can make ATP again, and then that ATP can again be broken down into ADP by releasing 1 phosphate. Okay, so when we are doing photosynthesis, basically we are converting ADP into ATP, so that later that ATP can be used for making glucose. Okay. On the other hand, when we do cellular respiration, we do use ADP and change it into ATP by using the energy from the glucose molecule. Okay. All right. So let's see our next step. So again, you should be able to answer these questions based on what we just learned. So let's talk about photosynthesis. Photosynthesis is when I think about photosynthesis, I break down this word into 2 words, photo and then synthesis. Okay, so photosynthesis actually does 2 things. One is the light part, the photo part, where the light energy is used to make ATP and to make NADPH. Okay, remember that another energy carrier that we have in that plant cells have. So that's the photo part, that you are using the light energy to make ATP. Okay, now the synthesis part is the Calvin cycle. Okay, CALVIN. So Calvin cycle. So so the plant cells under synthesis, they're making, synthesis means you're making something. So you're making glucose by doing the Calvin cycle, which is also called dark cycle, or it's also called the light independent cycle. So guys, again, the photosynthesis word has 2 things. One is photo, one is synthesis. Photo means that it is the light part of the photosynthesis, where light is used as a source of energy, and then synthesis is where you actually make glucose. Okay, now where does photosynthesis take place? Photosynthesis takes place in a special organelle which is inside plant cells, and that organelle is called chloroplast. Now chloroplast, if we look at the actual figure for a chloroplast, this is what chloroplast looks like. So you guys can see that this is an organelle, so it has a double membrane. So it has this membrane, just like cell has a cell membrane. These organelles for themselves have their own membranes. Okay, so those membranes inside actually have these structures that we call thylakoids. So these thylakoids look like, you can think of them as either pancakes, or think of them as coins stacked on top of each other. So these are the thylakoids. So thylakoids, guys, are the actual structure which contain chlorophyll molecules. Now, what is chlorophyll? So guys, chlorophyll is the actual pigment which is responsible for absorbing the energy from light. Okay, so again, I'll repeat the whole thing. You got your chloroplast, which is the organelle, inside which you have thylakoid, and inside the thylakoid you got your chlorophyll. So chlorophyll is the actual pigment responsible for absorbing the light energy. Okay, all right. So all right, now this, I think I already talked about this. So there's light reaction, and then there's light independent reaction, or Kelvin cycle, where sunlight and water will be used to make energy carriers and ATP and oxygen. And then in the second half of photosynthesis, we have Kelvin cycle, in which the carbon fixation takes place. What does that mean? Carbon fixation means you are turning carbon into a sugar. Okay, sugar basically means glucose. Okay, now where's the Kelvin cycle getting its energy from? Guys, the energy that is required for synthesis, or Kelvin cycle, is actually coming from the light reaction. Okay, light reaction produced ATP. That ATP is then consumed by our Kelvin cycle to fix the carbon into sugar. Okay, fixing carbon means you take carbon, like in the form of carbon dioxide, from the atmosphere, and that CO2, carbon dioxide, is changed into organic carbon, which is sugar, or we can call it glucose to be more specific. Okay, all right. So guys, this again slide is summarizing what I just said. Here's the light falling on your chloroplast thylakoids, which have chlorophyll, and then there are the one who will break down the water into oxygen and electrons, and then that will be used to make ATP and NADPH, which will then be used in your Kelvin cycle to change carbon dioxide into glucose. Okay, so this basically summarizes your two halves of the photosynthesis, the light reaction and the light independent reaction, or Kelvin cycle, or it's also called dark cycle. Okay, all right, now let's see in more detail what does that first half of photosynthesis look like. So that light dependent reaction, what's actually going on in there. Okay, so let me first so let's see here. You got these two regions that we are showing. So this is actually the picture of thylakoid, or you can think of chloroplast, and then this is you can see here, this is where the light is falling onto your photosystem 2. So guys, this is the membrane, and on the membrane you can see that there are some proteins that are situated. So these are called intermembrane proteins. They're sitting in the membrane. So you got this ATP synthase, you got this PS1, you got this another proton pump, and then we got this PS2. Okay, so PS stands for photosystem. Okay, this is the actual protein molecule which is responsible for absorbing the light. Now, guys, a very cool thing happens in photosynthesis. Okay, and what is that? It is this water, guys. What happens to water in photosynthesis? Now, plants, guys, have evolved to do this reaction, which is incredible, which is to take this water. You see water, a very stable molecule. It takes this water and splits that water into oxygen and H plus and this electron. Okay, now guys, if somebody is able to actually figure this out and do this outside the cell, if any scientist can figure out that how can you break down water and use it as a source of energy, guys, that person is ready to get a Nobel Prize, because this means that you can actually pump water into your car and run car using water. Okay, now that's incredible. So plants have managed to do that. Okay, so what do plants do? Plants use this light energy to break down this water into oxygen, H plus, and an electron. What does H plus means, guys? H plus also stands for proton. So if you guys remember from our second chapter where we learned the atoms and ions, so guys, hydrogen, if you take hydrogen and you remove its electron, it's going to break into H plus and one electron. So if you remove one electron from this hydrogen, it turns into H plus, and this guy is called proton. Okay, so wherever you see this H plus, it means protons. So so these protons are getting, you see there are lots of protons being created here. So guys, on the exam, if I ask you what is the source of protons in photosynthesis, the protons are coming from water, from H2O. Okay, so then the second interesting part is that we are making oxygen. Now guys, we breathe oxygen, and ever imagine where that oxygen is coming from? Of course, we know it's coming from plants, but what is happening in the plant? Again, photosynthesis is happening in the plant where the water is being split into oxygen. And this is the oxygen, guys, that we need for our cellular respiration. And that's why we breathe in oxygen. Okay, so and then the third interesting thing happening here is this electron that was made. This electron is considered a high energy electron. Okay, now what happens to this electron? This electron actually passes through this membrane, to this membrane space. So it goes through this protein. Now this protein is actually special. What does this protein do? Okay, see, this protein is also called a proton pump. Okay, why is it called a proton pump? This guy is able to pump the H plus ion into this area. Okay, so this region, guys, is called the lumen Okay, and guys, this part is called the stroma. Now guys, let me go back and show you where these places actually are. Okay, now if you guys remember, this was our thylakoid. Let me actually go back even more. So yeah, so this is your chloroplast structure, guys. So this is your thylakoid. So guys, this inside region of the thylakoid is called lumen. Oops, sorry. So this structure right here in the inside of the thylakoid is lumen. Okay, so this is where those protons were being pumped. So lots of H plus were being collected inside. And then where was it coming in from? It was coming in from this part. So guys, this is still chloroplast. Inside the chloroplast you have the thylakoids. Inside of thylakoid is lumen, but this portion, this part of the chloroplast is called stroma. Okay, so stroma is the region outside the thylakoid but inside the chloroplast. Okay, so now we'll come back to this figure. Okay, so we have, this is a proton pump. So this part again, as I said before, this is the stroma, so outside of the thylakoid, and this is the inside of the thylakoid. This is the lumen part. Okay, so protons are being brought inside, and there's a purpose why this is happening. So the goal of this electron transport, so this thing, guys, is called electron transport. Okay, this is also called electron transport chain, ETC. So what is happening here is electrons move, and that gives the energy to this proton pump to bring this H plus inside. And now you see we have accumulated lots of H plus in the lumen. So guys, this has created a chemical potential or electrochemical potential. What does that mean? So we got too much of H plus here, so lots of positive charge here and lots of H here, but here we got lesser H and we got lesser positive charge. So this has created a gradient. If you guys remember that from our previous lecture where we were talking about diffusion and osmosis, you guys remember that things always move from high concentration to the low concentration. So guys, right now we have spent the energy. Where's the energy coming from? From this electron, when the electron is transported. Okay, so that is bringing the H ion against the concentration gradient actively, because that required energy, and we accumulated H plus here. Okay, now what happens next? We'll later talk about H plus, what's happening with that H plus, but now what happened to that electron? That electron is then later transferred to photosystem number 1. Okay, so photosystem 1, guys, again is a protein complex which can now absorb more light. So there was light here which excited this water, and you got this excited electron. Now this electron does not have any more energy left, so more light falls in, and this electron is now changed into a higher energy electron. Okay, and now this higher energy electron, guys, is taken up by NADP plus. Okay, so what is this trying to say, guys? This is trying to say that if you take NADP plus and then you are adding H plus to it, and you are adding an electron to it, so guys, what will happen? You will make NADPH. Okay, and guys, if the question somewhere is asking you about NADP plus, it's different from you saying NADPH, because they are two different things. Okay, so what's happening here, guys? You see, you have NADP plus, and that guy actually gained an electron, and that's why you see this does not have any charge anymore. And that H is also now sticking onto NADP, and so you have made this NADPH. Okay, and guys, remember, what do we call this guy? This is energy carrier. Okay, this is not a direct ATP, but this will eventually give us ATP. Okay, I mean, especially when we talk about cellular respiration, when we talk about NADH, that's the one who's responsible for making ATP in the later steps. Okay, so now we got this NADPH. Okay, now important stuff, second important stuff, guys. What is happening with this H plus? Since we accumulated tons of H plus here, this H plus now goes back outside. Why? Because it goes from high concentration to the low concentration. So now what happened is your H plus is responsible for making this machine work. Now guys, this I would say so far is the most interesting protein molecule. What does this guy do, guys? Proton, when moves through this machine, through this ATP synthase, as the name suggests, produces ATP. So when you're producing ATP, what does that mean, guys? It means that you are using ADP. What does D mean? Diphosphate. So adenosine with 2 phosphates. And then you add a phosphate. I means inorganic phosphate. You add a phosphate and then you become ATP, adenosine triphosphate. That was the goal, guys, of doing photosynthesis, the photo part of the photosynthesis. So you have now made ATP. And again, how are you able to make ATP? It's because of this enzyme who uses H plus as its fuel. Okay, all right. So guys, what have we made in photosynthesis? We have made these two things. One is this NADPH, and then we have made ATP. Now there's actually, this is for energy side, right? But we have actually created one more interesting thing, which is this oxygen. Okay, so these three things are what are produced in photosynthesis. Okay, right, now we'll talk about the second part, the light independent reaction, or the synthesis part, or the Kelvin cycle, or the dark cycle. Now what happens here, guys? ATP, remember ATP from our previous slide? We have ATP and NADPH. So we have ATP and NADPH. Guys, they are the fuel for running this cycle, Kelvin cycle. Okay, and guys, Rubisco is actually the name of the enzyme who is responsible for, sorry about that, who's actually responsible for fixing or changing carbon dioxide into the sugar glucose. Okay, so you guys can see that ATP and NADPH are being consumed along with carbon dioxide to change carbon dioxide into sugar, and that's the purpose of doing Kelvin cycle. Okay, and this requires the enzyme called Rubisco. So it's ribulose bisphosphate carboxylase oxygenase. You don't have to remember the full name, but just know that you have an enzyme named Rubisco. Okay, all right. Now let's talk a little bit, since we just mentioned enzyme, let's talk a little bit more about the enzymes. What do we mean by enzymes? Again, how they work. So guys, enzymes are biological catalyst. What is that supposed to mean, guys? Catalyst makes the reaction go faster. Guys, there are two things about the enzyme. First, the enzyme will never participate in the reaction and get consumed. That will never happen. Enzymes are always recycled. Okay. The second thing is that they make the reaction go faster. That's the only thing they do. They don't make the impossible reaction become possible. They only make the reactions that are possible, make them go faster. That's what enzymes do. Now guys, most of the enzymes that we are going to deal with are made up of proteins, so basically made up of amino acids. Enzymes are very good at being very specific for their substrate. Now what is substrate supposed to mean, guys? So substrate is basically like a reactant, and then you're changing it into a product. Okay, so let's say you want to do this reaction where you take A, so that's your molecule number A, and then you got your molecule B, and then you want to change them into AB. So you want to join these two reactants and make this product AB. Okay, so this reaction is possible reaction, but if you add an enzyme to this reaction, it will make this reaction go much faster. Okay, so that's what the job is for the enzyme. And guys, enzyme are very specific for their substrate. So guys, A and B are the substrate for the enzyme. Okay, so and then AB is the product for this enzyme. Okay, now if we look at this figure now more carefully, yes, this is the actual enzyme, and you can see these are the two substrates like A and B. What happens here? Now this enzyme is very specific for these two guys. So these two guys come and sit on the enzyme, and now when that happens, you see the enzyme changes its shape. Okay, and it's called induced fit model. Okay, so what happens is these molecules do not just go and bind anywhere on the enzyme, but there is one site on the enzyme called active site, and that's the spot where the enzyme can tell, okay, this is the substrate that I want to bind to, and substrate, so enzyme has specificity to that substrate, so that substrate goes and binds here, and that changes the configuration of the shape of that enzyme, and now that enzyme does what? It changes the substrate, so A and B, into the product. Okay, so now you got that product, and that product is released. So this is also the product, but you can see this product is right now with the enzyme, and now then it's released, so taken off, and now you guys can see the enzyme goes back to its normal configuration, normal shape, and you can see the enzyme did not get consumed in this reaction, so the enzyme will then be used for running this reaction again. Okay, so what is the purpose of this, guys? So if you want to do 100 chemical reactions like these, changing AB to AB, I mean A plus B into AB, you don't need 100 enzymes, because that will be making too many proteins, too many enzymes inside our cell, and that's not a very not very wise to do, keeping in mind how much energy it takes to make proteins. Okay, so our cell is very wise in those cases. So the enzyme does not get consumed in a chemical reaction, okay, and it's recycled. Okay, right, now let's get into cellular respiration. What happens in cellular respiration? So now we're done with photosynthesis, guys. Now let's talk about cellular respiration, which we do and plants also do. Okay, now where does cellular respiration happen? Now if you guys remember, photosynthesis happens in an organelle called chloroplast. Now cellular respiration happens in an organelle called mitochondria. So mitochondria, guys, if you remember from the previous chapter, number 4, mitochondria is the powerhouse of our cell, and it's called that because cellular respiration takes place inside the mitochondria. So what happens again? The glucose, or complex molecule, is broken down into carbon dioxide and releases energy. Okay, all right, now let's see. Your cellular respiration, guys, consists of 3 steps. You got glycolysis, Krebs cycle, and oxidative phosphorylation. Now, guys, oxidative phosphorylation is also called electron transport chain, ETC. So we saw that ETC before. We just saw that in our photosynthesis as well. So, guys, oxidative phosphorylation, or your ETC, electron transport chain, happens in both respiration and in photosynthesis. Okay, now we think of these when we're studying them, especially the first time, it looks like glycolysis, which is the first step, happens first, and then it goes into Krebs cycle and then goes to oxidative phosphorylation. Like, it looks like a stepwise process, but actually all these 3 steps are happening in the cell at the same time. So if glycolysis has produced its products, they've already they go into the Krebs cycle, and that Krebs cycle was already using the glycolysis product from the previous glycolysis that happened. So all the 3 steps are actually happening at the same time, but for our convenience, we study them one at a time. Okay, so this figure is summarizing all the 3 steps. So, guys, on the top you see glycolysis. So glycolysis is basically taking this glucose and breaking it down into pyruvate. So, guys, if you see, glucose has how many carbons? It has 1, 2, 3, 4, 5, and 6 carbons. Now it's broken down into 3 and 3 carbon 2 molecules. So 2 molecules of 3 and 3 carbons, and that molecule is called pyruvate. Okay, so you guys should know that for each of these, so like for glycolysis, for Krebs cycle, for oxidative phosphorylation, what is used and what is made. Okay, so for glycolysis, glucose comes in. This glucose is first charged with ATP, and then there are many steps, guys, happening between going from glucose to pyruvate, but for this intro class we just talk about glucose and it gives us pyruvate. We keep it that simple. But when you go to your advanced courses, you will see that there's actually lots of steps that are happening when you go from glucose to pyruvate. So on net basis, glucose, when it's changed to pyruvate, so a big molecule broken down into two small molecules. First of all, guys, just by listening to that, does this look like an anabolic reaction or a catabolic reaction? This is going to be a catabolic reaction because you're taking a big molecule, breaking into two small molecules, okay? This not just makes pyruvate, but also makes ATP and NADH, okay? So guys, ATP is the direct energy currency that we need for our cell to work, but it also makes NADH, which is not bad, because NADH is like our asset, like gold. We're going to use it to sell it when we need, and then we can get cash from there, and for our cell, NADH can be used to make ATP. And we're going to see where that happens. That actually happens here, right here in electron transport chain, the oxidative phosphorylation. That NADH is used to make more ATP. Okay, so we'll get there in a second. Now, it's very important, guys, to realize where is what in the cell. Okay, where's what is happening. All these chemical reactions are happening at a different spot, and we are responsible for knowing where everything is happening. Okay, so you got this cell structure, it's a whole cell. This is your mitochondria, this is your nucleus, these are your endoplasmic reticulum, you got your Golgi apparatus. Now, this portion of the cell, so guys, you got these organelles, and that's a cell, and in between that you got cytoplasm. So cytoplasm is like the actual place where all these organelles are sitting. So this fluid-like structure, this place, this is called cytoplasm. So this is cytoplasm right here, okay? So glycolysis takes place in the cytoplasm, okay? Now, when we get to the Krebs cycle, this Krebs cycles actually happen inside the matrix of mitochondria. So guys, this is mitochondria, and mitochondria is also special, like chloroplast. Mitochondria actually has two membranes, okay? And it's important, guys, for you to understand where's what. Let me see if there's a bigger figure for mitochondria. No, there's not. So we'll stick to this one. So here you guys can see we got the outer membrane. Oops. Okay, so we got this outer membrane. So this right here, this is the outer membrane of mitochondria. And then you see this one, the one which is twisted. This is the inner mitochondria. Okay, right. Now you see this place, this yellow portion, yellow light color, this is called the matrix. So this is like the inside of mitochondria. Okay, guys, expect on your exam and your study quiz, you should be able to label each one of these. Okay, so again, outer membrane, inner membrane. What do you think this place should be called? This place is called the intermembrane space. So this is the space between the membrane. So intermembrane space. Okay, it's very important to understand what do we mean by each of these. Okay, right. So where's Krebs cycle happening, guys? This pyruvate that was made in cytoplasm, because glycolysis happened in cytoplasm, this pyruvate needs to be moved inside the mitochondria for a Krebs cycle to happen. So this is where the Krebs cycle happens, guys. In the matrix, that's where Krebs cycle is happening. Now, where does the next step, which is the oxidative phosphorylation, happens? As it happens on the membrane. On the inner membrane, you got to this that you're seeing here. That's your inner membrane. That's where all the machines are to do this process of converting NADH and FADH2 into ATP. Okay. All right. Now, what happens in the Krebs cycle, guys? Krebs cycle. The word Krebs is the name of the guy who first discovered that reaction like this happens, and he actually got a Nobel Prize for this because this was a very important discovery that happened. Now, you can see pyruvate. So this pyruvate, two pyruvates were made. one at a time goes into the Krebs cycle. Guys, this Krebs cycle is also called citric acid cycle because the very first product that is made using the pyruvate is actually citric acid. Okay, that's why this cycle is also called citric acid cycle. Now, you guys are not responsible for knowing exactly what molecules are participating, but guys, this is actually 8 steps of reactions that happen in Krebs cycle. Okay, so you had pyruvate. Now guys, pyruvate has how many carbons? Each pyruvate has 3 carbons. So in one cycle, 1 carbon dioxide is taken off. Okay, so you can see what is happening to the glucose carbons. You can see in cellular respiration, this glucose carbon will ultimately be changed into carbon dioxide. Okay, and that's why this is a catabolic process, right? You're taking a big molecule into small CO2. An exact opposite, guys, happened in photosynthesis. If you remember synthesis part, CO2 was used to make glucose. Now it's opposite. Okay, now is the CO2 the only thing made? Of course not. The thing that we're trying to make here is this NADH. That's one thing which is made, and actually 6 of them are made. Okay, 6 of NADH are made. Then 2 ATP are made, and then FADH2 is made. I think 2 of them are made. Okay. All right. So that's when you put in 2 pyruvates. Okay. Now you got these ATP and you got ATP here. These are the ATPs that you can directly consume. I mean, our body can directly use. But you can see we have also collected this NADH from glycolysis. We have collected NADH in mitochondria because of the Krebs cycle, and we have also produced FADH2 again because of Krebs cycle in the mitochondria. Okay, this guy on the other hand, this NADH was made in cytoplasm and was brought into the mitochondria. Okay, now what's happening here? So again, to know what is where, this is the matrix. And guys, what is matrix? Matrix is the inside of the mitochondria. And then what is this place? Guys, this place is the intermembrane space. Okay, so if you consider this to be the inside of the mitochondria, which is matrix, now we are throwing out the H+. You can see H+ are being collected in the intermembrane space. So this space right here between the 2 membranes. So this is very similar, guys, to photosynthesis electron transport chain. That's where also ATP was being made by moving the electrons through. Okay, so guys, remember NADH and FADH2, these 2 molecules that are being made here, they are being used to transport their high energy electrons. And guys, those electrons are used by these proteins. These proteins are also proton pumps. What do? They take protons from here and bring them to the intermembrane space. So what we have done is we have collected tons of H+. Okay, now what happens to this H+? The lots of H+s that we have collected here, they go back again through the ATP synthase, very similar to photosynthesis. And now what happens? ADP is changed to ATP. So guys, in cellular respiration, you see you have finally made ATP from where? From this NADH. Remember the gold that you can go and sell to get dollars? You can use this NADH to spend its electron, and in exchange send the H+ here and then make ATP. Okay, so this is how NADH can be changed to ATP by doing oxidative phosphorylation or electron transport chain. Okay, now let's see what happens to this electron. Now you guys know that we need oxygen, right, for respiration. So guys, we breathe oxygen. Yes, we need oxygen, but why do we need oxygen? Because you can see in all reactions so far for cellular respiration, we have not used oxygen. Okay, now this is the last step, guys, of oxidative phosphorylation, and this is where oxygen is directly required. Okay, so oxygen is the acceptor of this electron. So this electron that went through all these different proteins finally goes to oxygen and makes water. Okay, so guys, in photosynthesis, oxygen was produced. Okay, and that oxygen in cellular respiration is being used as a final electron acceptor to create water. Okay, so oxygen is actually consumed in cellular respiration to make water. Okay, of course now you have produced ATP, and that's the whole point of doing glycolysis, Krebs cycle, and this last electron transport chain. Okay, now it's very important to understand, guys, that all these things, guys, are happening in our cell all the time, and that's how you're making ATP. But what happens if there's no oxygen? So when there's no oxygen, does that ever happen? Yes, guys. Imagine when you're exercising and your body has limited oxygen. What does our body do then? Or imagine, guys, you are in a forest and then you have a bear chasing you. If you have a bear chasing you, do you want to wait for this ATP? Your body's gonna make this ATP, and eventually you will start running. Do you want to depend on this? Because see, this one is taking time. NADH was made in these 2 steps, and then it took all this electron transport chain, pumped the protons, ATP synthesis makes ATP. Do you want to wait till then to run? No, of course not. What happens then, guys? We want this ATP. Okay, so guys, when you're exercising or you're running, this is the ATP that you're dependent on. So glycolysis is your first source of ATP. And guys, bacteria who do not have mitochondria, they also depend on glycolysis to get their ATP. Okay, all right. Now, so I think this is just summarizing what I just talked about. Now you can see oxidative phosphorylation requires oxygen. So guys, if I say which step and which step does not require oxygen, this is the only step in cellular respiration which does not require oxygen. Without oxygen, you can keep doing glycolysis. You will be able to run Krebs cycle for some time, but eventually it's going to stop. Why is it going to stop, guys? Think about this. You see, if you have no oxygen, this is not going to run because you got no oxygen. This will for sure stop working. Let me open the pen. So if you got no oxygen, this whole thing stops. You are not making this ATP. You're not moving these electrons. So guys, if you stop taking these electrons from NADH, you are stuck with NADH. You see what is happening here? NADH is being changed to NAD+. So if you do not have oxygen, you will keep accumulating NADH. So guys, if you keep accumulating this NADH and you do not have any NAD+, if you do not have any FAD, do you think you can run Krebs cycle? No, you cannot, because NAD+ is required to run the Krebs cycle. So if you do not have oxygen, it of course affects electron transport chain, but also affects your Krebs cycle because your NAD+ is not available anymore. Okay, so you can only then do glycolysis, make this ATP, and then feel pain in your muscles. Now, why did I say you'll feel pain in your muscles? So guys, when you're exercising, running, you must have realized that if you work too much, then your body starts hurting because we are making lactic acid. Okay, and lactic acid is made because of the fermentation. Now guys, why do we need fermentation? It's very important to do fermentation. Okay, now there are 2 types of fermentations. One is to make alcohol by doing photosynthesis and carbon dioxide. The second is to make lactic acid. Which one do you think we do? We do the second one. Which one do you think yeast does? Or the ones that are used for making alcohol from grapes or any other source? That's alcohol. They're the one who do alcohol fermentation. Okay, so guys, remember your glycolysis. Your glycolysis change your sugar into pyruvate. Now we have to take care of this pyruvate, right? Now there are 2 ways to take care of it. One is to go this way, lactic acid fermentation. That's what we do. The second is to go alcoholic fermentation, and that's what yeast does. Okay, now what happens here? You take the pyruvate and change that to lactic acid. What has that done? What has that accomplished? Why do we need to do this, guys? We need to do this because this changes NADH to NAD+. Now, what is great about that, guys? See, if you change NADH to NAD+, do you need NAD+ for running glycolysis? Yes. For glycolysis, you do need NAD+, even if you want to make this ATP. Okay, so who changes this NADH back to NAD+? It is this lactic acid fermentation which is going to change this NADH back to NAD+. So guys, this NAD+ is then required to do glycolysis more and more, and you can keep running. Okay. Now, yeast, what does yeast do? They do alcoholic fermentation, so they take the same pyruvate but change it into ethanol and CO2. Ethanol, guys, is 2 carbons. Pyruvate was 3 carbons, as you can see. So that 1 extra carbon ends up being carbon dioxide. Now, same thing happens to NADH, change to NAD+, so solves the same purpose of making more NAD+, but this one makes it ethanol, this one makes lactic acid. Okay, you guys ever wonder what will happen if you start doing this? If you start doing the ethanol fermentation, what's going to happen is you're exercising and you're making ethanol. That will actually make you feel dizzy. It's going to make you feel as if you were drunk, right? Because you're making ethanol. Imagine how that would be, okay? But we, of course, don't do this. We don't make ethanol. We instead make lactic acid, and that hurts us, okay? And then that's when we stop exercising, okay? All right, so that is it, guys, for your chapter number 4. I hope you guys liked the video. Let me know, guys, if you have any comments about this video. You can email me and give me any recommendations on how you would want your videos to be. Okay, all right, see you guys next time.